Hello class, thank you all for joining me today. In this lesson, we will go over the nucleic acid, which is the final macromolecule we will be talking about. Now, just to recap, so we have an idea of what we're talking about as the lesson goes on. These last 4 lessons have been about the macromolecules. They consist of monomers and polymers. Macromolecules are made up of polymers, which are in turn made up of monomers. In our last lesson, we discussed proteins. Proteins are made up of the polymer peptides, or peptide links, which are in turn made up of the monomer amino acids. Amino acids, in turn, are made up of carbon-based structures. In the same vein, we have the nucleic acid. The polymer of the nucleic acid is the DNA and RNA strand. And the monomer of the DNA and RNA strands are what we call nucleotides, which we'll go into more detail as we go on. So just to recap, we're talking about the macromolecule nucleic acids, which are made up of the polymers DNA and or RNA, which are in turn made up of the monomers of nucleotides. It's also worth mentioning that while not completely accurate, a very simple way is that DNA is 2 strands of these polymers, while the RNA is 1. Essentially, you can think of RNA as half a strand of DNA. That's not quite the way that it works, but that is a very simple way of thinking about it. So let's get on to it. Nucleic acids are the most important macromolecules for the continuity of life. They carry the cell's genetic blueprint and carry instructions for its functioning. The 2 main types of nucleic acids are deoxyribonucleic acid, or DNA, and ribonucleic acid, RNA, and I'll not be saying those full names again. DNA is the genetic material in all living organisms, blah blah blah. Essentially, DNA is the blueprint, while RNA is like the copy machine, if you will. You have DNA, which is basically how your organismness, if you will, is how your body and bodily structures function, while RNA is what reads it to make another cell, or in a when you were a fetus, a zygote, the simple way to build you the first time. Other types of nucleic acid, RNA, is mostly involved in protein synthesis. As we discussed earlier in proteins, I believe we did, I think we went into that detail. Proteins is one of the most, is basically your basic structures. Very often when your proteins are out of whack, then a lot of your functions don't work properly. That's why RNA is so important. DNA and RNA are comprised of monomers that scientists call nucleotides. The nucleotides combine with each other to form a polynucleotide, DNA or RNA, or polymer. 3 components comprise each nucleotide: a nitrogenous base, which basically means a base that is centered around nitrogen, a pentose, which means a 5-carbon sugar, and a phosphate group, which it'll be a lot easier to explain what that is once we get a photo, which I'm pretty sure we will. Each nitrogenous base in a nucleotide is attached to a sugar molecule, which is attached to 1 or more phosphate groups. And here we go. The most common one that you hear a lot in nutrition or organic chemistry or microbiology, whatever, will be the pyrimides, or how to pronounce it, and purines. I could never pronounce that correctly. Now you're probably asking, what's the difference? Well, the big difference is the number of carbon rings. Here, the purines have 2, and the pyrimides have 3. These letters are going to be important because what separates RNA and DNA is going to be the difference in the sequencing of the monomers. For instance, all RNA and DNA have your adenine, guanine, cytosine. However, DNA only has thymine, and RNA only has uracil. DNA will never have uracil, RNA will never have thymine. And this is why I said that thinking about RNA as half of DNA is not quite right, because it's structurally a little different, but essentially as you will have the RNA be double helix, in other words, two strands of DNA wrapped around each other, but RNA will only be one helix. In other words, it's going to be itself wrapped around itself. I'm hoping that this thing has good photos, and that'll make a lot more sense. The nitrogenous base, important component of nucleotides, are organic molecules that are so named because they contain carbon and nitrogen. Each nucleotide in DNA contains one of the four possible nitrogenous bases: adenine, guanine, cytosine, and thymine. Scientists classify adenine and guanine as purines. The purines, I think is how it's pronounced. Primary structure is two carbon nitrogen rings. Scientists classify cytosine and thymine and uracil as pyrimidines, which have a single carbon nitrogen ring as their primary structure. The pentose sugar in DNA is deoxyribose, and in RNA, the sugar is ribose. The photos. Okay, on. Oh, here we go. Deoxyribose and ribose, and the reason they call it that is because on the ends, you have in DNA you have the deoxyrization, I guess it'd be oxidation, or I don't remember the word. I think we discussed it a couple of videos back about removing oxygen. What was that called? Dehydrating or something like that? Well, anyway, as you can see in the deoxyribose, you're missing an O on the opposite side of your two main chains. But here you have it. So that's the main difference. Anyway, the difference between the sugars is the presence of a hydroxyl group on the ribose's second carbon and hydrogen on the deoxyribose's second carbon. The phosphate residue attaches to the hydroxyl group of the 5 carbon of one or 5th carbon of one sugar and the hydroxyl group of the 3rd carbon of another sugar in the next nucleotide, which forms the 5,3 phosphodiester linkage. A simple dehydration reaction, like the other linkages connecting monomers in macromolecules, does not form the phosphodiester linkage. What this basically means is, here's DNA. In fact, it's not a great photo, but it'll do. Here is a DNA structure. You can see, like, you have one strand. If you look, you can see one strand here, another strand here, wrapping around itself. RNA will only have one of these strands. And what that means is, with each little connection, it's either it's connected to the 5 to another 3, and then that 5 to another 3, etc., etc. That's what this means. DNA has a double helix structure. The sugar and phosphate lie on the outside of the helix, forming the DNA's backbone. That's what they call it. This thick colored line is the DNA backbone. These remain the same for every DNA strand. Like me and you have different DNA structures and sequences, but our DNA backbone is the same. It has to be the same to function properly. Hydrogen bonds bind the pairs to each other. Every base pair in the double helix is separated from each base pair by 0.34 nanometers, which I believe is what NM is. The helix's two strands run in opposite directions, meaning that the 5th carbon end in one strand will face the 3rd carbon end of its matching strand. And the science calls it anti-parallel. So basically, on these two color lines, if the 5 is up here and the 3 is up here, the 3 will be up here and the 5 will be down here. Only certain types of base pairings are allowed. For example, a certain purine can only pair with a certain pyrimidine. This means that A can pair with T and G can pair with C, or in RNA, A can pair with U. And the reason for that, I believe we will go through this next several lessons later, but that essentially means, the reason is that there's something in A and G, or A and C, that repel each other. So A can only combine with T and G can only combine with C. Essentially, see these two that are kind of connected, they're attracted to each other? Well, if this is adenine, this has to be thymine. Or if this is cytosine, this has to be guanine. It cannot be, like, this cannot be adenine and this cannot be cytosine. It just can't happen. This is the base complementary rule. In other words, the DNA strands are complementary to each other. If the sequence of one strand is AATTGGCC, the complementary strand has to be TTAACCGG. And here's actually a very good way of look. In fact, here's a good photo of how it operates. Essentially, the nitrogen, if you recall, I don't even have my periodic table, but I think off the top of my head, how many does nitrogen have? Oh man, I forgot. I think it has like 5 valence electrons. I think, I don't remember, but it has the right number where it can share the hydrogen, which keeps it attracted. And O has 1, I believe. So that's how these things are being able to attract to each other. A mutation occurs and adenine replaces cytosine. What impact do you think will have on DNA structure? We'll get into mutations, I believe, in genetics. As mutations, essentially, physically, and quite literally, is when the strand doesn't work the way it should. Is when, like, guanine is connected to adenine. Ribonucleic acid, or RNA, is mainly involved in the process of protein synthesis under the direction of DNA. So RNA is the copy machine, and DNA would be the blueprint. RNA can also be considered the builder, as DNA is the boss or something. Like I've heard that one before. RNA is usually single-stranded and is comprised of ribonucleotides that are linked by phosphodiester bonds. There are 4 major types of RNA: messenger RNA, or mRNA, ribosomal RNA, or rRNA, transfer RNA, or tRNA, and microRNA, miRNA. I don't think we go over microRNA all that much. That is not something I remember going over. At the first, mRNA carries a message from DNA, which controls all the cellular activities in a cell. If a cell requires synthesis in a certain protein, the gene for this product turns on, and the messenger RNA synthesizes in the nucleus. The RNA base sequence is complementary to DNA's code sequence for which it has been copied. However, in RNA, the base T is absent and U is present instead. If the DNA strand has a sequence of AATTGCGC, the sequence of the complementary RNA is UUAAGCGCG. In the cytoplasm, the mRNA interacts with ribosomes and other cellular machinery. Ribosomal RNA, or rRNA, is a major constituent of ribosomes on which the mRNA binds. The rRNA ensures the proper alignment of the mRNA and the ribosomes. Ribosomes, which we'll go over a little more, or have gone over. We have gone over in the chapter with the cells, and we'll go over a little more detail when we really get into inside the cell. But ribosomes are inside the cytoplasm of a cell that interact with numerous things. So that's what ribosomal RNA is. It combines the mRNA and the ribosome inside the cell, basically linking them together to help synthesize protein inside the cell. Transfer RNA, or tRNA, is one of the smallest of the 4 RNAs, usually 70 to 90 nucleotides long, which is not very long at all when you compare to how long pretty much anything with a chain is. It carries the correct amino acid to the protein synthesis site. It is the base pairing between the tRNA and mRNA that allows for the correct amino acid to insert itself into the polypeptide chain. And here is a great table of how things function, and it's we're actually comparing. Okay, so it's comparing DNA to RNA. DNA carries genetic information, while RNA involves in protein synthesis, or basically copying off the DNA. DNA is always in the nucleus, but RNA can leave the nucleus. If DNA leaves the nucleus, the cell either breaks down or does not function properly. This is basically the brain of the cell. And here you go. So, yeah, screenshot this or whatever. This little thing could be very useful in any test or any memorization you want to do. Even though the RNA is single-stranded, most RNA types show extensive intramolecular base pairing between complementary sequences, creating a predictable three-dimensional structure essential for their function. As you have learned, information flow in an organism takes place from DNA to RNA to protein. DNA dictates the structure of mRNA in a process scientists call transcription, and RNA dictates the protein structure in a process scientists call translation. This is a central dogma of life. That is important. The central dogma of life states that the DNA, the source of DNA, the DNA, when that's created, it sends out the RNA, which copies the DNA, and the RNA goes out and creates the proteins for other cells. That's why I call it the central dogma of life. If that doesn't happen, life cannot exist. It just can't. So every time something is rebuilt in your body, new anything is rebuilt, it starts with this. Your existing DNA, which is created when you were a zygote. The RNA for that DNA copies your original DNA code, transports it up to other places to create proteins. So cell synthesis can begin over and over again. We call that mitosis. Oh, I guess that was a short chapter. Yeah. So guys, I guess that's it. Now, I asked those last few videos, but I have not gotten any kind of answer. Do you guys want to do any of the review questions? We can do them together. Maybe help you think about how. Maybe help you get thinking on these questions. And if you want to, please let me know. I would think about doing the multiple choice, but if we want to do these critical think questions, we can do that as well. So please let me know in the comments. The very next video we will do, the very next lesson we will do, will be the cell structure, beginning chapter 4. This is where it starts to get really interesting. Like when I was talking earlier about ribosomes, we spoke about that before, but now in the cell structure we will get into a lot of that stuff. Trust me, the cell structure is very interesting and very important, regardless of what kind of biology you're getting into. While cell structure for me is important because I want to become a microbiologist, virologist, even if you're a macrobiologist or whatever, this is going to be very important to know and very interesting. So I just want to please let me know in the comments if you guys want to go over some example questions, and that's it. So I just want to thank you all very much for watching. Have a wonderful day, and God bless.